Thanks, CC, for that introduction. And I just want to start by thanking the organizers for the invitation to speak here today. I'm really excited to be here and to speak after all the great talks earlier today, especially the ones today about personalized medicine as it applies to oncology. I think there's a lot of common themes there with what I'll talk about. My talk will be a little bit different, but I think a lot of the same themes apply. So what I'm going to talk about is tell the whole story kind of from our initial hypothesis and discovery of a compound all the way through kind of taking it through using canine cancer patients. These are pet dogs with cancer that we use to treat and to help these animals, and also that we use to advance our compounds, and then through to basically the cusp of clinical trials. So the talk will necessarily be technical in some of the details on the discovery of the compound and its mechanism of action, but I think there'll be a lot of general lessons here about drug discovery process and how one thinks about a novel target and moving compounds forward, and really getting through some of the very difficult things, the so-called valley of death, where it's very difficult to advance some basic discoveries and translate them to the clinic. So just as a disclosure, and as I'll talk about later, we formed a company called Vanquish Oncology around this compound, and I am a co-founder of that company. So some of the objectives are, we're going to talk about the apoptotic pathway and how one can impinge upon that pathway in terms of novel drug discovery. And for this specific target and compound, we're talking about a compound that activates an enzyme called Procaspase-3. This is a key linchpin in the apoptotic cascade. And also the use of pet dogs with cancer, client-owned dogs. These are patients from the community. And using these animals that are very different than advancing compounds through mouse models, which is the typical way that things are done, where mice are given cancer and then treated with compounds. These are veterinary cancer patients that we're using to provide humane treatment for these animals, but also to evaluate our experimental therapeutics. Okay, so most anti-cancer drugs are really accurately classified as anti-proliferative agents. These are compounds that arrest rapidly dividing cells in different phases of the cell cycle. So compounds like cisplatin or cyclophosphamide, DNA-damaging agents, S-phase arresters, antimitotics such as Taxol and vincristine arrest in the M phase. And so, as I mentioned, these are general cytotoxins, and they induce death in all rapidly cycling cell types. So, of course, these have been life-saving drugs for thousands of patients a year, but they do have dose-limiting toxicity and, in many cases, not a lot of efficacy against metastatic disease. And so if one thinks about personalized medicine and as it applies to oncology, there's a nice review about 10 years ago talking about where good targets come from. And some of these are themes that we've heard at different sessions today. Good targets come from, say, translocation. So the poster child for this, imatinib or Gleevec. This is a reciprocal chromosome translocation between chromosome 9 and chromosome 22, resulting in the so-called BCR-ABL fusion protein. And so now this is a target that's more akin to an antibacterial target. This target is only really present in the cancer cells. And it's been mentioned, this compound and this drug has been transformative for patients with this translocation, specifically CML. Another good personalized strategy or personalized target strategy for developing a personalized medicine is a mutation. So there's a compound, gefetinib, Iressa, that is very specific for a certain mutation in patients that have this mutation in the EGFR. So if they have this mutation, this compound is an excellent inhibitor of this mutant enzyme and not affecting other enzymes. And again, for people with this mutation, the compound is highly effective. Another target is overexpression. So these are situations, you can have binary overexpression where you have say, expression only in the cancer cell and not in the tumors, only in the cancer cell, not in the normal cell. So that's more akin to something like a translocation again. Or there can be differential expression between the cancer cell and the non-cancer cell. And this has been demonstrated by drugs like Herceptin. Okay, so something we think a lot about in my lab is the apoptotic cascade and how to impinge on this for drug discovery. And about 99.9% of your cells will die through this apoptotic cascade. Two major pathways through the intrinsic and extrinsic pathway. Okay, and these both converge to the activation of this enzyme, this proenzyme protease, Procaspase-3, that gets activated to caspase-3. Once caspase-3 is activated, this is a cysteine protease that cleaves 100 plus substrates in the cell. So one of the hallmarks of apoptosis is the evasion. One of the hallmarks of cancer is the evasion of apoptosis. And so cancer cells do this through many different mechanisms, through, say, the upregulation of anti-apoptotic proteins such as BCL2, methylation of APAF, the expression of decoy receptors. All of these result in a net break of this circuit. So if you think of the apoptotic cascade as a circuit between, say, an extracellular receptor and the ultimate effector, this circuit is broken in cancer cells, and so this allows the cells to continue to proliferate and become a tumor. And so our hypothesis a number of years ago, and this is kind of the key conceptual slide for the talk, was that we could identify a compound that directly induces the autoactivation of Procaspase-3 to caspase-3. So if we could identify a drug that did this, we could induce death in cancer cells even that had aberrations and alterations in their apoptotic circuitry. So this could be a very powerful therapeutic strategy. Another advantage of this strategy and a benefit of directly activating Procaspase-3 is, as I mentioned, both of these apoptotic pathways, the intrinsic and the extrinsic funnel to the activation of Procaspase-3. These mutations, as I mentioned, are above the Procaspase-3, but also, as it turns out, Procaspase-3 levels are elevated in certain cancers. And so this is a study here from a group at Merck where they looked at normal tumor from different cancer patients. And as you can see from this Western blot, they found that Procaspase-3 levels were elevated 6-fold in colorectal cancer. Subsequent to this, many people have identified elevation of Procaspase-3 to hold for many different types of cancer. So what it looks like is in these cancer cells, there's this enzyme that could kill the cell that does not get activated, but it is actually elevated in these cancer cells. So if one can come in with a small molecule and activate this pool of proenzyme protease, one can get specificity for a cancer cell over a non-cancerous cell. Okay, so just briefly about the biochemistry of Procaspase-3. So Procaspase-3 itself is an active enzyme. It's about 200-fold less active than caspase-3. And Procaspase-3 can auto-process itself, so it can cleave itself to the active caspase-3. So our vision was if we can enhance Procaspase-3 activity, it could cleave itself, cleave another molecule of Procaspase-3 to caspase-3. Once caspase-3 is activated, it can go back and cleave, say, another molecule of Procaspase-3 and activate this apoptotic cell death pathway. Okay, so this was our goal, to find activators of this proenzyme protease Procaspase-3. And so what we did was to set up conceptually, I think, very simple screen where we can recombinantly express and purify this enzyme Procaspase-3. We have a collection of small molecules. At the time we did this, we had a modest-sized collection of about 20,000 compounds that we either synthesized, had purchased, or had collected from the attic of the chemistry building at the University of Illinois. And we simply incubate this enzyme with these compounds in the wells of a 384 well plate using these simple pin transfer devices, and then add this caspase-3 substrate. Any compound that allows the processing of this caspase-3 substrate is an active hit. So through this process, a number of years ago, we published on this compound that we call PAC-1, or Procaspase Activating Compound 1. So chemically, this is an orthohydroxy-N-acyl hydrazone. This is a stable compound, as I'll show you, both in buffer, in lysate, and also in multiple animals with a elongated half-life in these animals. And so what I'm going to talk to you about today is the mode of action. So it's unusual that most pharmacological agents and drugs that you know of are enzyme inhibitors. And this has been proven to be a very successful strategy in the pharmaceutical industry. So this is a little bit different, and an enzyme activator. So it's important for us to determine how does a small molecule, how does a drug activate an enzyme. Also, through an outstanding collaboration with the veterinary medicine folks at the University of Illinois, we've been able to evaluate these compounds in pet dogs with cancer. And this has really been transformative for us in terms of both identifying optimal compounds to move forward with, but also to optimize dosing, formulation, and we found this compound to be orally bioavailable. PAC-1, as I'll show you, is blood-brain barrier penetrant, and we're really excited about it as a potential agent for CNS cancers, both primary and metastatic. And also PAC-1 synergizes with multiple anti-cancer drugs. So how does PAC-1 activate Procaspase-3? So if you'd asked me at the beginning, when we discovered, I probably would have said it works by an allosteric activation mechanism where it binds to some site away from the active site. Another possibility, however, was that it inhibits the inhibitor. This is a theme in biology where there'll be an active enzyme and an inhibitor, and then something else comes to relieve that inhibition. So these are kind of the hypotheses that we set out to address. And so one of the things that we discovered early on was by making chemical derivatives of PAC-1, we identified compounds that were inactive. Okay, these compounds do not kill cancer cells in culture or activate Procaspase-3 in vitro. And by looking at the functional groups we deleted, we could identify the functional groups that are important for activity. In a similar fashion, we found that we could change the periphery of this molecule, the two sides of this, if you will, and retain activity. And so we focused in on this, what's called this orthohydroxy N-acyl hydrazone as key, really, for compounds that had this, were active Procaspase-3 activators and also active inducers of apoptotic cell death. And so we began to suspect metal binding. We knew that this orthohydroxy N-acyl hydrazone is a metal binding motif. And so we knew from the literature that zinc inhibits caspase-3. And so we did some of our own experiments to show that zinc will also inhibit Procaspase-3. Remember how I said Procaspase-3 has this low level of protease activity, and we showed that zinc inhibits Procaspase-3. We also showed by isothermal titration calorimetry that zinc will bind to Procaspase-3. So it binds to Procaspase-3 with a relatively modest affinity of a dissociation constant of about 350 nanomolar. So through these experiments, we developed the hypothesis that, and asked the question, does PAC-1 activate Procaspase-3 in vitro via zinc chelation? So basically, chelating zinc away from Procaspase-3 and allowing Procaspase-3 to be active. And does it work by the same mechanism in cell culture and in vivo, inducing death via the relief of inhibitory zinc from Procaspase-3? So we were able to very quickly show that PAC-1 and zinc bind to one another just through simple titration experiments. We can show that PAC-1 and zinc bind with a dissociation constant of about 50 nanomolar, so tighter than the interaction between zinc and Procaspase-3. We also showed that PAC-1 will relieve zinc-mediated inhibition of Procaspase-3. So this is Procaspase-3 by itself without any zinc. This is with the addition of zinc, and as we titrate in PAC-1, we restore Procaspase-3 activity. So consistent with the notion that PAC-1 is chelating this labile inhibitory zinc. So this is our hypothesis about how PAC-1 works. So Procaspase-3 has been shown by others will co-localize in the cell with zinc. PAC-1 with a slightly stronger affinity can come in and chelate zinc from Procaspase-3, enabling Procaspase-3 to activate itself and induce cell death. So there's good evidence from others that PAC-1 works this way. 10% of zinc exists in labile loosely bound pools, so about 90% of cellular zinc exists in very tightly bound zinc stores, matrix metalloproteases, zinc fingers, et cetera. We've shown that PAC-1 does not affect those enzymes. But these labile zinc pools, PAC-1 can chelate. And more recently, the inhibitory site on Procaspase-3, caspase-3 has been identified. So the zinc binding site on Procaspase-3 has been identified by others. We did an interesting experiment to show that this mechanism is operational in cells using genetically encoded zinc sensors. This was through a collaboration with Professor Amy Palmer at the University of Colorado, where she has developed these zinc sensors, a fusion of CFP and YFP and a zinc binding domain. So when zinc binds, you see a FRET signal that connects these two. So this is kind of data that one gets from these experiments where with resting cells, and you can flood in then a zinc chelator, and then flood back in zinc, and you see a restoration of the signal. And so the kind of traces one gets, T-pen is a very strong zinc chelator. So once you add that, you see a diminution of the FRET signal, and then you can add zinc back. The nice thing about this is these sensors are highly specific for zinc over the other relevant metals. And so we thought this was a great way to probe this phenomena and our hypothesis in cancer cells in culture. And so in combination with the Palmer lab, we added PAC-1 to cells that express these sensors. And so immediately you see a diminution of the signal consistent with chelation of this labile zinc. And you can add more. Importantly, when you add a very strong zinc chelator such as T-pen, it will then start chelating zinc promiscuously and pulling zinc out of other tightly bound other proteins where zinc is tightly bound. So PAC-1 will only chelate this labile zinc pool. So our conclusion from this experiment was that PAC-1 chelates this labile zinc pool inside cancer cells. In the 10 years or so since this compound has been disclosed, there's been a lot of studies from others that have correlated and confirmed our data. This is a really nice experiment that was published earlier this year by another group where they showed that PAC-1 will activate Procaspase-3 before the other caspases. If you recall from the apoptotic cascade, Procaspase-8 and 9 are first activated in kind of canonical apoptosis, leading then to the activation of Procaspase-3, and that's what most standard pro-apoptotic agents will do. With PAC-1, we see activation of Procaspase-3 first, and then one sees activation of Procaspase-8 and Procaspase-9 several hours later. Again, consistent with the idea that this compound is directly activating Procaspase-3 in the cell. The compound has selectivity, again, as shown by others, for cancer cells, killing cancer cells versus non-cancerous cells, not having much of an effect on non-cancer cells in cell culture. And these people also showed confirmed the zinc dependence and efficacy in multiple mouse models. There's also a very nice validation of and use of PAC-1 as a tool to study Procaspase-3 activation by a group here in this manuscript, and others have shown that PAC-1 has no effect, again, on true zinc proteins, where you have a zinc site that's critical for the catalytic function of the protein. PAC-1 is not inhibiting or pulling zinc out of those enzymes. Okay, so when faced now with a potential anti-cancer compound, there's a lot of directions that one can go. And so, you know, the kinds of things that we think of are the types of cancer, what types of cancer can we apply this to? Efficacy, how can we demonstrate real meaningful efficacy in sophisticated tumor models? And recognizing that most cancer drugs now are developed and will be applied as drug cocktails, can we show and does the compound have efficacy in combination with standard of care agents? And then really getting through this valley of death, and how can one actually move this compound and in general move compounds through that and towards a human clinical trial. Okay, so one of the things that we discovered pretty early about this compound is that it's blood-brain barrier penetrant. And this is very unusual. Most anti-cancer drugs do not get into the brain. And this is why metastatic disease to the brain, to the CNS, and primary CNS tumors such as glioblastoma are very, very difficult to treat. And so we have a prediction algorithm that we use that takes into account some different factors, polar surface area and C log P. And we predicted from this that PAC-1 would have about a 30-70 partitioning ratio between the brain and blood. And just to calibrate you, temozolomide, which is used for glioblastoma, is about 15 to 85. So that's considered a very penetrant drug. We designed a compound that we didn't think would get in at all, SPAC1, and this, again, according to our prediction algorithm, would not get in. When we actually did the experiment and treated mice, sacrificed, and determined the levels in the brain and the blood, we found that these predictions were pretty close. For PAC-1, the partitioning is about 30 to 70, and SPAC1 is a compound that really is almost entirely excluded from the CNS. So this suggested to us the potential of this compound PAC-1 for CNS tumors. So we wanted to have kind of parallel development pathways a compound for CNS cancers, PAC-1, and then a compound for systemic tumors, non-CNS cancers, this compound SPAC-1. What we found was that PAC-1, in our initial studies when we were giving it IV at very, very high doses, it caused this hyperexcitation, which also suggested, again, that the compound was penetrating the blood-brain barrier. But this caused us initially to turn to this compound SPAC-1 as a very safe compound that we could evaluate dosing strategies and pharmacokinetics in various comparative tumor models. Okay, so I think one of the things that we've talked about today in this conference is the cost of drugs and the cost of cancer drugs, and of course it's extraordinarily high, and part of this is because of the failure rate of drugs. So even compounds that start a phase 1 clinical trial in oncology, depending on the stats that you look at, as many as 19 out of 20 compounds that start a phase 1 trial will not make it to become drugs. So this is a very bad success rate, and so why do oncology candidates fail? There's a lot of reasons, of course, but one of the big ones is the negatives of the mouse xenograft model. So typically, drugs are discovered and evaluated in mice, in mouse models of cancer, contrived models where mice are given cancer and then treated. And although there are improvements in these models, and typically there will be homogeneous cell types and non-metastatic disease. And so we've been asking ourselves, what about dogs? Dogs are about 25% of all dogs will die from cancer. So this is about 1 million patients per year in the US alone. These are obviously relatively large and outbred compared to mice. They often have not received other chemotherapeutic treatment. And so for us, this is a great opportunity to assess our compounds in real patients, veterinary cancer patients with spontaneous tumors. So it's interesting that many of the cancers that dogs get are very, very similar to the human disease. For example, non-Hodgkin's lymphoma, if you're a human and you get this, you'll be treated with CHOP therapy, or in some cases R-CHOP therapy. Dogs also get treated with CHOP therapy. Molecular signature profiling has been done looking at the genetic similarity between human and canine cancer. So this is transcript profiling on canine tumors and human tumors and their cell line counterparts. And what these studies have shown is that this is for metastatic osteosarcoma. These studies have shown that the human and canine osteosarcomas are not distinguishable from one another based on their global genetic expression signature. So they have the same mutations in the apoptotic pathway, P53, BCL2, et cetera. Okay, so this was the first animal that received our compound. This was SPAC-1, where at this point we were giving this IV, and we can have a catheter where we're able to monitor levels of the compound in the blood. So Red is the name of this dog, and it's hooked up to an IV. This is a IV pump here. Red is hooked up. This was a 24-hour continuous IV infusion. And so here is the pharmacokinetic results, and you can see with the IV infusion, we see a spike and then a leveling off. And we can achieve very, very high doses, so over about 30 micromolar for over 20 hours in the blood. So this compound, SPAC-1, safely achieves very high serum concentrations for 24 hours. So this led us to do a clinical trial of SPAC-1 in lymphoma dogs. And so the way this works is these dogs are enrolled in cohorts of 3. These are either previously treated with CHOP therapy or drug naive. This is a very aggressive disease in dogs. So untreated, these tumors will double in size, and the dogs will die in about 4 to 6 weeks. So the way that they measure outcome is through the RECIST method, the addition of the longest linear lengths of the 8 peripheral lymph nodes, and we do CT scans as well. So in this particular case, the treatment was SPAC-1 continuous IV infusion once a week for 4 weeks. And so we want to confirm safety and pharmacokinetics and look for any efficacy. And so this was our first patient. This is my collaborator, Tim Phan. This was Zombie. This dog is a dog from the community that was in remission after CHOP therapy, but the cancer had returned, as it always does. And so Zombie was treated once a week for 4 weeks with SPAC-1. And so this is Zombie in the CT scan. This is the treatment schedule for Zombie. So what we saw was a shrinkage of the tumor size in this single agent study with SPAC-1 upon treatment of just 4 treatments over once a week for 4 weeks. After we took Zombie off the study, the tumors regrew. These are 6 dogs that we did in this initial phase 1 study. And these are, again, dogs. These are all comers from dogs with lymphoma from the community. We saw 1 dog with tumor regression, 3 with stable disease, and 2 with tumor progression. So this was very encouraging in this initial study of SPAC1 in cancer patients. What we subsequently discovered is the parent compound PAC-1 given to dogs in a capsule shows outstanding pharmacokinetic properties. So what Tim did was to put the compound in a capsule and then just put it in a meatball. The dogs will gobble it up, and it actually shows outstanding pharmacokinetic properties. So what we avoid with this is that big spike that one gets from IV, which was causing the neuro excitation, and we see this nice, smooth pharmacokinetic curve, high levels of the compound when given orally, and dramatically reduced CNS hyperexcitation, and we don't see it in these animals. So this is obviously a much more convenient and translatable mode of drug delivery. We've made a lot of PAC-1 derivatives, so SPAC1 is a compound I mentioned to you. Other groups, this compound is fairly easy to manufacture, so other groups have made this compound, and now over 1,000 derivatives have been made. We made 800 in a study shown here. There's a compound that we call BPAC1 that is moving forward for hematological malignancies. A Chinese group made this very interesting compound that really showed a lot of potential, I think, compared head-to-head with PAC-1. It's a big molecule, so it is not blood-brain barrier penetrant. So, over 1,000 compounds have been made, and so far compounds with the tolerability, efficacy, and blood-brain barrier penetrance, the combination of all 3, superior to PAC-1 have not been found. So we moved forward with PAC-1 itself. And we wanted to move forward with this into glioblastoma. And so just quickly, over 1,200 Americans each year are diagnosed with this disease. This accounts for about 2% of US cancer deaths. Untreated, the median survival is 3 months. Radiation was the standard of care for a long time, and this extends survival to about 12 months. In 2005, there's a landmark phase 3 trial of temozolomide, this drug here, TMZ plus radiation, and this was found to extend survival by 2.5 months. So this was a large phase 3 study, and with this trial, radiation plus TMZ became the new standard of care for glioblastoma. Obviously, still a lot of room for improvement for treatment of this disease. So what we found is that PAC-1 can be given in mini-capsules to rats. So you can barely see these here. These are very small capsules that we can fill with the compound and give them to rats. So our collaborators at Johns Hopkins, Professor Greg Riggins, has a rat model of glioma. So this is an intracranial model. So the compound has to penetrate the CNS to have activity in this model. And we showed that PAC-1 as a single agent, given as the oral solid, extends survival very dramatically in this 9L glioma model. What we thought was that based on the mode of action, based on this zinc chelation and activation of Procaspase-3, was that PAC-1 could synergize with a lot of other drugs that work through the apoptotic pathway. So we thought that by taking the brake off of Procaspase-3, we could induce apoptotic death individually with PAC-1, but also in a magnified way, we could induce apoptotic death in combination with standard of care anti-cancer drugs. And so we've been exploring that with temozolomide. So this is just some of our cell death data with different glioma cell lines. The dashed line here is the additive effect. So we see synergistic, very nice synergistic cell death. That synergy is also shown in apoptosis. So this is PAC-1 on its own, the apoptotic quadrants. This is TMZ on its own. At these concentrations, it has very little effect. But you can see when they're combined, it has an effect greater than the additive effect. And you can see this by Western blot as well. Activation of Procaspase-3, PAC-1 on its own, TMZ on its own, and then the combination of the two, we see activation of Procaspase-3 and cleavage of caspase-3 substrates. So our collaborators have been doing these animal models with PAC-1 and TMZ, and in this case, they did PAC-1, just 5 doses of PAC-1 at a level that we knew would not have much effect on its own, plus TMZ, and then the combination. Again, in this very short dosing, we see an extension of survival in this rat glioma model. From this, they moved to a more sophisticated mouse model of glioma where human oncospheres are intracranially implanted, and PAC-1 and TMZ, again, are given. And this goes for close to 300 days. And again, based on this, PAC-1 alone, TMZ alone, and the combination shows dramatically extended survival when these two compounds are used together and synergize to extend survival of these animals. Okay, so an interesting thing is the allometric scaling. So this is conversion of an animal dose to a human dose. So just because rats and mice are given 50 mg per kg does not mean that is the equivalent human dose. And if you're interested in this, I really advise you to read this paper where they talk about this. So a PAC-1 shows efficacy in mice and rats at 50 mg per kg. 50 mg per kg is, after allometric scaling, is equivalent to a human dose of 8 mg per kg for a rat 50 mg per kg dose. A mouse 50 mg per kg dose is equal to a human dose of 4 mg per kg. So these are in dose ranges that are very, very attainable. Okay, so to finish up here, moving towards human clinical trials. So about 3 years ago, we founded a company called Vanquish Oncology to move PAC-1 into human clinical trials. Last year, about 18 months ago, we got an angel investment, about $4 million, that enabled what we were able to do, expedited IND-enabling studies. So there's very strict prescribed studies from the FDA that are required before one can submit an IND to do a phase 1 clinical trial. So we were able to do these in a rapid way. So chemical manufacturing, so manufacturing the compound on a large scale, and formulation. Acute and chronic toxicity in rats and dogs, effect on cardiac, blood pressure, and respiratory in dogs. And then we did all of this, and then we're able to file our IND just this past month. And so this was all able to be done because we were very, very confident in the safety of this compound based on the dozens of dogs, pet dogs with cancer that we have treated with this compound. There's a kilogram scale synthesis of PAC-1, Regis Technologies in Chicago. We outsourced this to them. What they were able to do was they got quantitative yields, so 100% yields on all of these reactions to get to PAC-1, 75% yield to get to this half, and then the combination of these two, we get to the active pharmaceutical ingredient. This has been made on a 10,000 gram scale. These are the pills. These are 250 mg tablets of PAC-1. And this is a bag of about 300 pills. Okay, so the way that this is going to work, the phase 1 clinical trial will take place. Component 1 will be at the Cancer Center in Chicago, and this is a classic dose escalation in late-stage cancer patients. Dose escalation to MTD once a day for 21 days. There's a component 2 that we are hoping to do at Johns Hopkins with glioblastoma patients. So this is a combination of PAC-1 plus TMZ in glioblastoma patients, and this is where we think we hopefully can help these patients in this very, very difficult to treat disease. So the way this will work, this is the standard treatment for glioblastoma, where you have TMZ plus radiation, and then adjuvant TMZ therapy and these different cycles. So basically what we're going to do is just superimpose PAC-1 on this stage, where we will then administer PAC-1 in this phase once a day for 21 days. And again, this will be a dose escalation study of PAC-1 in combination with TMZ until the MTD is reached. So in conclusion, what I hope to have told you about today was this novel target that we have been thinking about, a novel strategy. That is the direct activation of Procaspase-3 as an anti-cancer strategy. PAC-1 is a compound that does this, and it does this by this unusual mechanism of sequestering inhibitory zinc ions from this proenzyme protease. We're able to use pet dogs with cancer. These are animals that really do not have other treatment options, are often euthanized. We're able to offer them this experimental therapeutic, and now we've treated dozens of animals with PAC-1 or SPAC-1 in combination with different therapeutics. The compound can be given orally and is safe at high doses. The compound readily penetrates the blood-brain barrier and has efficacy in these different CNS models. It shows synergy with a wide variety of standard of care cancer drugs, and this is something we're really excited about, is once we kind of define the safety as a single agent through our phase 1 trial, to explore this, and we've been doing this in preclinical models, but to explore this with different cancer drugs in patients in very difficult to treat cancers. So the direct procaspase activation is a novel anti-cancer strategy, and really the use of pet dogs to expedite and to speed drug development and to help these canine cancers patients that don't have other treatment options are some of the take-home messages that we've gotten from this and things that we are doing with other programs. So again, thanks a lot for the invitation to speak here. Thanks for your attention. I'd be happy to answer any questions.
Okay, Paul, I gotta ask. How'd you come up with the idea to use dogs?
Well, it really, my veterinary collaborator, Professor Tim Fan, contacted me after seeing our paper, and he is a veterinary oncologist, so he treats cancer patients from the community, and he, his hands are tied in many ways. They can't use drugs that are on patent; it's simply too expensive. And so they're left with off-patent drugs. And for many of these cancers, such as the ones I mentioned, there really isn't a good treatment. And so he wanted to be able to offer his patients something. And also, for us, it was a great way to be able to assess, because as I mentioned, part of the cost in this whole business is compounds that go through but then fail, right? And so for us, the idea was if we could identify compounds that are going to have tox problems or lack of efficacy early on, and only advance the compounds that we really think are active in this model, which I think is much more challenging than rodent models of cancer.
And I think you're starting to get at a couple of the questions that have come in from the audience while you're speaking. And one was specifically, are you using the pets just for the tox studies or?
No.
No.
No. So these are client-owned dogs. These are pet dogs that have cancer, and so the veterinary oncologist's intention is to treat those animals. Of course, we're getting PK data. Of course, we're getting tox data. But this was a phase 1 study, and things are moving forward in that direction as well.
Okay, good. A couple of maybe a little more scientific stories here. One has to do with what other types of cancers you see with these high levels of Procaspase?
Caspase, yeah.
Caspase, hard for me to say, that might be appropriate for this PAC-1 therapy.
There's lots of them. So in our initial paper, we showed colorectal cancer, colon cancer, lung cancer, breast cancer, all have elevated levels of Procaspase-3 that have been documented by other people in other studies. And so those are ones that we're really looking at in preclinical models. Metastatic breast cancer is one that we're looking at. There's no real standard of care for that. There's a lot of different single agents that are used. And so we think that's an opportunity where we can combine PAC-1 with some of these and hopefully see enhanced efficacy.
And so that makes me just wonder, when you're thinking about sort of next targets of opportunity, is that going to be your first line criteria, or in your mind are there maybe a couple things that you'll look for?
Well, so obviously expression of Procaspase-3, the target, is a top criteria. There's a lot of cancers, as I mentioned, that express that. I think our next one is really unmet medical needs. So that was really a big driver to go into glioblastoma. Even though it's a very difficult disease to treat, we thought we had the potential to help these patients, hopefully, because the compound is somewhat unique as a cancer agent as it gets through the blood-brain barrier. So I think other diseases, as I mentioned, diseases that metastasize to the brain, that has the same challenges, although in some of those cases, the blood-brain barrier may be compromised and drugs can get in, but I think in many cases, the blood-brain barrier remains a major obstacle to treatment of patients with CNS mets.
A good one here from the audience. How are normal cells protected from being sent into the apoptosis?
By this compound?
Okay.
Yeah,
walk us through that a little bit.
Right. So normal cells have that apoptotic cascade that I showed you. So there's the so-called extrinsic through the death receptors and intrinsic through the mitochondria. And so they will receive signals, and at certain times when they're no longer needed or when they're precancerous, those are identified, shuttled off to the death pathway. As I mentioned, 99.9% of our cells die that way, and normally that's a very good thing. In the case of this compound, how are they protected? We believe it's because of the differential level of expression of Procaspase-3 in the tumor versus the normal. So in some of our own studies with lung cancer biopsies and colon cancer biopsies, it can be 6 to 20-fold elevated in these patients. So if the compound comes in, it's going to be much more effective in activating Procaspase-3 in the cancerous cell than the normal cell.
Okay, so I'm not sure who sent this in, but it's clearly somebody in the audience who knows I could hit it out of the park. But I'm gonna let you try this question, because obviously, why did you use zinc in acidic form and salt form? Why not salt in both cases? Obviously, Simple question for me to answer, but you give it a try, Paul.
Well, I mean, in a buffered solution, the zinc will form ions and bind to this inhibitory site on Procaspase-3. So I think depending on the pH, I don't know that the form that we use matters. We've used zinc chloride, we've used zinc sulfate, and we've shown that PAC-1 will work on both of them. And then also in the cells, which I think with those genetically encoded zinc sensors, we can show that we're mobilizing this labile zinc pool in the cell as well.
So I think you've given us some really nice optimism here, thinking about PAC-1. It certainly does seem promising from your presentation, but I do think getting back to sort of one of the important themes of this gathering is sort of the practical application. Do you have a sense of how far off we are from getting this into clinical setting?
So we filed IND in September, and so based on our discussions with the FDA, it's very likely to start the clinical trial before the end of the year.
Okay.
So
all right.
that's, I mean, it's a frustrating process. It's slow, but with a new compound, they're very, very conservative, unfortunately. But I mean, it's fortunate in some ways, but unfortunate for the cancer patients. But the good news is, of course, that these phase 1 trials are in late-stage cancer patients, and then there's a rapid dose escalation. And so we hope to be at dose levels that we think will be active at the early part of next year.
Okay. Well, on that note, let's say thank you to Paul.